अब हम बात करने जा रहे हैं लार्ज स्केल आइसोलेशन ऑफ प्लाज्मिड डीएनए, जिसको मैक्सी प्रेप के नाम से भी जाना जाता है। इससे पहले हम मिनी प्रेप के हवाले से बात कर चुके हैं। मिनी प्रेप और मैक्सी प्रेप में जो फ़र्क है वो एट द एंड अमाउंट ऑफ डीएनए आइसोलेटेड में है। मिनी प्रेप डील्स विद द सैंपल्स फ्रॉम वेयर वी गेट micrograms of DNA, whereas maxi prep is a procedure by which we can isolate milligrams amount of DNA. There are different methods which can be employed for maxi prep, but in all the methods the steps are more or less similar. What we need for maxi prep is the similar as we talked in mini prep. For example, we need growth of microorganism as a source of DNA or the plasmid DNA, harvesting of these microorganisms, then lysis of the cells to separate or to isolate the DNA, and then purification of the plasmid DNA from cell debris, which contains the proteins, particles or parts of the broken membranes and the genomic DNA. In general, the bacterial growth and lysis follows the same pattern. We grow bacteria in the same manner as we did for the mini prep, and we can use the same bacteria for maxi prep. However, to yield the high quantities of plasmid DNA, we should ensure that the bacterial culture have reached to a saturation level. The lysis of the bacteria after the saturation level is achieved and after the harvesting is done, the lysis of the bacterial cell can be done using the mixture of mild detergent and alkali solution that is going to release the compact plasmid DNA. Most of the larger DNA, which is normally remained bound with the plasma membrane, will be separated as part of the cellular debris. Why the plasmid DNA is not affected by alkaline treatment? This is because of the fact that the plasmid is a covalently closed circular DNA and it has no response, or it is not affected by the alkali which is used in the lysis buffer. Once lysis is done, now this mixture should be processed further for purification of the plasmid DNA. We have different methods established in the labs to isolate or to purify the plasmid DNA, the most common of which is cesium chloride method. This is the traditional method which is used in the labs for separation, purification of the plasmid DNA. However, nowadays this method is not preferred to be used in the laboratories because of the reason that it involves the use of toxic chemicals like ethidium bromide or the organic solvents like phenol. After the modification or after the advancement in the molecular sciences, the columns have been developed which are known as affinity columns. These columns have affinity to bind to charged particles because they contain some resins, and while using these resin containing columns, we can easily isolate we can easily purify the plasmid DNA from the cell suspension, from the lysed cells. Such columns, one of the example is anion exchange resin. As we know that the DNA is negatively charged molecule, it has close affinity with positively charged particles. So these columns carry the positively charged particles to which negatively charged DNA can bound, and later on it can be eluted or purified using the gravity-based chromatography procedure. This procedure can be performed relatively quickly because of as compared to cesium chloride method. As far as purity of the plasmid is concerned, this gives the purified plasmid equivalent or greater than the cesium chloride method.